these genes. So as a summary, there is an increasing interest in a mixed population because they represent an opportunity to identify variants of different ancestral origin and frequency that may contribute to phenotypic variation. There is a high diversity of European, Native American, and African ancestral proportions. There is a significant stratification and socio-spatial segregation of these proportions. There is a different geographic distribution, and this complexity of the distribution of the genetic components in the population is both a challenge and an opportunity to understand the role of genetic or genes in association with different biological traits.
Any questions?
The thing you're saying about Yucatan, couldn't that be explained also with some type of migration process, or is that not possible? Do you think it's just a sample bias?
Yeah, I think this is sample bias because actually, when we look at the individuals in our sample that came from Yucatan, they're all from Merida and from high socioeconomic status. So it's very likely that actually the bias is because people from the rural areas in Yucatan are not, they are very, less likely to sort of migrate to Mexico City to study. I think that's
right. You also mentioned about why you didn't find significance for the correct. Is it possible it could also be like interactions between genes? Can also be a possibility you didn't find like one specific gene coding for it?
interactions genes. Well, yes, I mean it's possible, but it's very difficult at this point to model the interaction.
You're already comparing so many genes, so
Yeah.
you include
And
interaction.
actually, there are not too many models. Well, yes, there are some computational models to look at interactions. But yeah, so basically, I think that the next step is to look at the relevant lifestyle factors that different people with the part of the sample with higher European ancestry, higher Native American.
What can you tell us about this gene related to that predispose people to obesity too? In particular, Mexicans. It has been very popular in the news.
But which one, FTO or?
Yeah.
Yeah. Well, actually, we did a study like 5 years ago with FTO and different samples in Mexico. I mean, but this is not specific of the Mexican population. It's the variant, the allele risk is common in Europe and different regions. It's not particularly, it's not more frequent in the Mexican population.
Yeah.
So it looks like it's common in our population. So it's not useful to understand the particular susceptibility of the Mexicans. If that exists.
Just a quick question. So these plots, you were showing all the variability across all the different samples from Native Americans in Mexico.
No, it's the Mexican sample in general.
So across everything.
Yeah.
So,
So it's the 1,600.
So I wonder if you partition the variance from the different samples across Mexico. So we know the native population in Yucatan and native one in Mexico. So presumably it might be the case that those people might have get some correlation in the genotype, and then that variation can be masked by putting it all together.
Yeah, I think this is a good idea to split the database, taking into account different geographic origin or socioeconomic status. We already included socioeconomic status, geographic origin as covariates. So here these analyses are corrected for covariates as demography, origin, also age, sex, other variables that can be relevant. But yeah, it's a very good one. Actually,
Well, next we have Janet. Gonna present landmarks and spatial orientation of the brain.
So I'm going to talk about a different type of maps, not so much from Mexico, but this is one that lives within our brains. So I'm studying a PhD in neuroscience at UCL, and I'm trying to investigate the relationship between how is it that our brain encodes space and how that maps to the external environment. So at first, I don't know, maybe some of you are local to UCL, some come from external places. And one of the ways that you navigate is basically by translating a map with some reference system, depending on where you are located and where you are heading. So there are different components that you need to integrate to basically find your way around. So this is a sketch of the UCL main campus. Imagine that you arrive from Houston Station, and so you look at this map, and so you basically have to decompose information. So you can use a coordinate system, more or less, to determine where you are and where you want to go. And then from there, you can estimate the relative distance. But as you are actively moving, you also have to find out where you are in relation to landmarks, because otherwise you'll get constantly lost. So imagine if there's an environment where everything is the same, and you can only rely on your self-motion. But using features of the environment, you kind of know where you are heading and in which direction. And that's important because our sense of direction is an integration of multiple processes. So it uses external information from the environment, your self-motion, and also where you are facing at the moment and where you are heading. So this can be studied in the rodent system, and you can break down all that information that I showed in the map into what these animals might use when they navigate. So as I said, they can use this estimates derived from their own motion. They can also use information from the environment, either the distal horizon, and also the proximity of local features that the animal knows, like the home range, or in this case, if it's a rat foraging outside of your garden, maybe the fence. So all this comes together in a way that the animal has a very good mapping system of where it's being, where it's going, and the positioning of all those locations. And that is translated into some amazing behavior. So this is the grand whistling rat in the sub-Saharan desert. And it has such a good mapping system that it lives in subterranean holes, and there are about 150 different entries. So whenever there's a potential predator, the animal can compute its direction, its current direction, to where one of these locations is, and then basically escape from the predator. Or if there's an animal that might enter the burrow system, the animal can map the short escape. So it has an internal map of space within the brain. So the question is, how does that actually, how can we measure that? And how can we investigate it? And early on, a lot of psychologists tried to answer that question with primarily cognitive tasks, which was training the animal to do a particular behavior. And they thought that the way the animal understood the world, that the way that they map the little universe, was based on associations. And John O'Keefe was a professor at UCL and won the Nobel Prize last year in medicine. He thought a different approach. He said, why don't we just put the animals in an environment, let it move freely, instead of being trained in any task, let's just try to relate how the activity of neurons correlates to where the animal is located in space. So the idea that he had is that the animals have this internal representation system that will be found in a region of the brain. And there were previous studies, actually in humans, where they found that lesions in the hippocampus will lead to these severe deficits in finding your way around in navigation and also in memory, which is tightly related to the spatial representation of space. So the way he approached is he developed, along with others, this technique where you record with very thin electrodes, positioning particular brain structures, you record the activity of single neurons, and then later you can isolate that, you can decompose that. And on the top of the rat's skull, there's a set of LEDs that allows you to, with a camera overhead, to track its position. So then you can relate the activity of neurons, shown here in each of these dots. So every dot is one time that the neuron fired, one spike. And then here in the black lines is the path of the animal, so the route that it took as it was moving around. So you can see that that these cells don't fire just randomly. They seem to fire at particular locations. And here's a heat map, kind of like what Victor showed for the distribution of Native Americans, where it shows the firing rate of the cell, where the peak rate is in red, and the blue colors is where the cell firing rate is. So you can see even here that there seems to be some information that the animal is encoding about particular locations, right? The important thing about this is that it's recorded in while the animal moves and just throw pellets around. If you turn off the light, this activity remains. So that tells you that this is a representation of the animal's location independent of the external environment, so that it can be maintained by its own self-movement. And that's important, and I'll bring that issue a bit later. So along with John O'Keefe, this couple in Norway, they co-shared the Nobel Prize for finding what became known as grid cells. So grid cells are basically these amazing single cells that fire at very periodic locations arranged in hexagons. The way that they came about with this idea is they said, okay, if there are many place cells that encode local locations in the environment, how is this all come together? There must be a binding process that brings all these different places so that the animal maps all the environment. So they went and record in a location that was highly interconnected with the hippocampus called the medial entorhinal cortex, and that's where they found these periodic cells. So the question is, what do they actually represent? What do they tell the animal? So they basically tell the animal location as place cells, but also distance, and distance in relation to boundaries. There's also a third spatial map in the brain, and these are so-called head direction cells. Unfortunately, these were not awarded the Nobel Prize, but these are the ones that I happen to work with. So these are recorded throughout many regions, originally found in the post-oviculum, and they have a very, very interesting feature, is that they fire more or less like a compass. So again, this is a plot of the animal just moving around, and whenever the cell fired one spike, you can see in blue dot, and then you see these like burst or a high firing rate when the animal is facing northeast. Here in the other plot, you can see that the animal is just dwelling along the path, and then here's the number of times that it fired a spike. You can see that there's a huge activity when the animal happens to be facing here, southeast. So this tells the animal where it is in relation to the environment, but also primarily where it is in relation to where it's facing. And again, just as with place cells and grid cells, if there's no light in the environment, if there's no available external landmarks, the activity can be maintained in darkness. So its own self-motion can tell the animal where it is with a little bit of error. And I can show that in the next slide. So you can quantify the activity of these cells by making what are known as tuning curves. And tuning curves is just a distribution of the firing rate of the cell and the heading direction of the animal, which is obviously 360 degrees. Another way of visualizing those tuning curves is just with in polar coordinates. And then here I just superimposed to show that, that the cell had a high firing rate when the animal was facing in that direction. So then, as I said, these cells can be maintained in darkness, but the activity of external landmarks can also tell the animal where it is facing. So the way that you can relate to this is, imagine that you come into this room and there's absolutely nothing. It's just an empty room. If someone was to disorient you, like you did with piñatas, you can find your bearings. So it's very difficult to know what you're facing. So then you can rely on your self-motion to know where you are. However, if there is any available visual information, then you automatically say, okay, now I know what I'm facing because that's a familiar. Say the door. Say that this is all just white rooms, and then okay, so that's the door, so that must be the direction in which the exit might be. So then your system, your internal system, can basically translate that activity and shift in relation to that information. If this is not clear, then I will probably stop because this builds, what I do builds on this. So are there any questions about how?
So visual landmarks will adjust, calibrate.
Exactly.
Send
Yeah,
directions.
exactly. They calibrate it such that if you turn off the lights, the activity of these cells will drift a little bit, change, and then once you turn on the lights again, activity is reset.
But if there's no landmarks or lines or any cue, it just it's based on movement.
Exactly. It's based on its self-motion. So that was shown in the early studies, where what they did is an experiment where they just placed the animal inside a cylinder with one cue and curtains around just to minimize the information, the external information, and have this as the most salient cue. And they rotate the cue by taking the animal out of the environment, placing it back, and so on. And that's a cue manipulation because you don't fool the animal just as much as you don't fool a human. So if you were in this room and all of a sudden you see that the door changes, you say, okay, that's not reliable. However, if I take you out of the room, disorient you, you don't know exactly where you are, and then the whole thing changes, say the door is now in that direction, then your internal coordinate system would align to that.
Yeah. So that's a circular arena.
Yeah.
What about if you change the shape
Okay.
to, rectangular, like an hexagonal or?
Okay.
Does that
Yeah. Can I answer that question towards the very end because the answer is has many levels. So the geometry does influence, but on specific circumstances. Okay. So then here's just an example that cues control the activity of all the spatially modulated cells: the direction cells, the phase cells, and the grid cells. So I focus on the head direction cells because two factors: they have a really clear signal to noise, and also you can characterize by one feature, by where the cell fires, so you can map that activity to how it relates to the external world, to the cues. Yes. What, 5 minutes? Whoa, I think I can go fast. Okay, so I focus on two brain areas that basically bring the input to the spatial map, and these are the retrosphenial cortex and the postsubiculum. And just previous studies have shown that if you lesion these areas, the postsubiculum or the retrosphenial cortex, you get an effect such that when you rotate cues, the cells don't follow. So then they have an important role in the processing of landmarks, which doesn't happen obviously in the control. When you change the cue, you have a really good mapping. So the overall research question is interesting: what visual features drive the activity of these head direction cells? And to do that, I do a variant of the standard task in a cylinder with two cues that vary on a specific feature, and that feature varies along different dimensions. So it could be contrast, orientation, and then here's another orientation point with a different polarity. And then these are cut off here, but these are just mirror images of each other. And this one's a bar looking high and above, and that's just the identical cues as control. So then the hypothesis is that if the animal can map the location of these two cues and use them as landmarks, if it can tell those two apart, then the cell should maintain its direction or its relative direction relative to those cues, right? Like I said, to the door you are aligned, and your mapping system is just it tells you where you are facing. However, if that's not the case, then it should alternate between the two cues. It should switch by 180 degrees because that's the nature of the task. So then that's the expected outcome if you record a population of neurons. If there's absolutely no relationship, then the distribution should be normal. If it's a strong control by the cues, it should be unimodal. And if it's not the case, it should be bimodal, just switch between the two cues. How am I doing on time? 2 minutes? Okay, so you need to quantify the activity of these cells in relation to how these cues are rotated. So first you need to filter out what constitutes a cell that is tuned to head direction. And then later, because I do a series of sessions, I need to find out a way to quantify how much the cells shift in relation to those cues. So how one thing maps to the other. And that's basically what I show here is that I can compute the angle from where the cell fires in relation to that particular cue. Okay, so because this is circular data, it gets a bit tricky. You can't treat it as linear. So you want to show how all those plots are distributed along all the facing directions. So there's a way, this friend of mine, who will be here unfortunately again, he's a statistician, has helped me and said that, like, one of the best methods that you can use to show this is basically to do a density estimate. And density estimates won't go into details if you're interested. But if you have a lot of data, then you can compute the distribution, that there's particular clustering along that continuous variable. So these are the results. I recorded 124 of those interaction cells, 17 rats in 2 brain regions, 12, 5, and so on. So these are the results. So remember the hypothesis and the model. It said that if the animal can tell these 2 QO parts and use them as landmarks, we should expect to see strong bimodality, unimodality, and almost grouping at 0, right? So then you can see that most of the cells tend to be clustered around that location, telling us that those 2 Qs can be highly discriminated. And then with the bars, quite similar result. And then with those, not much data here compared to the other ones, but still a nice effect with the vertical and horizontal bars. And then with the high and low bars, notice that these distributions are becoming more bimodal. And then with the symmetric Qs, they continue to be bimodal. And then interestingly, when I did the control, I thought that it would become bimodal, meaning that just switching between those 2 Qs, but it turned out that now the animal uses the difference between the black Q and the background of the cylinder. So it's completely symmetric, and the animal doesn't care much about these 2 Qs. They can use any available information from within the cylinder. So this is just to show that there was no difference between the 2 brain areas, and that's important because theoretically, there have been models that show that that all the activity of these cells is tightly correlated, tightly coupled. Time? Yeah. All right. So then once so we've seen all the distributions, and there's supposed to be a way now to classify the behavior and test it. So so I created categories based on previous results, where it showed that what is the maximum likelihood or the maximum range that a cell can change in relation to just 1 Q, and they showed that that was within plus and 45 degrees. So then I split the distributions into categories, and I assume that those cells that fell within this category, category 1, would be the ones that that corresponded to having a good mapping system between the Qs. Those that change are those that flicker between the 2 Qs, so the animal can't tell the information apart. And then these in between, I can't say much about it, except that perhaps it's using some feature of the cylinder, so the intramace white, sorry, the gray color. Okay, so then because the samples were a bit different, it's biased to just compare the frequencies. So with the Bayes analysis, you can generate a distribution based on the frequencies and then compare that data. So then you can compute the probability and estimate how likely is it that an event will fall in one or the other categories. And these are just 2 examples of the posterior distributions from that. And here you can see, so I put the 2 extreme examples, from one that has the best Q control and those that have very different ones. So just quickly, if we just look at the probability of data falling within 1, meaning that there is a really good Q control. Notice that if you look more or less about the median here, probability is about 0.80, whereas here it's much lower. So then these analyses, what they show is that if I was to record many rats, many cells, the likelihood of finding an animal or a cell that would use those Qs and landmarks would fall within some probability. So that gives me more power with the data that I have. And that just is a mapping of, well, not a mapping, but it's just a distribution or an interval for each of these categories, just showing that that for the likelihood of falling within a category, it varies across the Qs, more or less what you saw in the distributions. So summary, time. All right. So summary. So I've hopefully showed that these edge reaction cells can maintain its internal activity, and they also use some features of the environment. In these cases, they're low-level visual features, such as contrast, orientation, and so on. And also that the likelihood depends on the properties of that stimulus. So the greater the difference between the two, the more likely it is that the animal, the cell, will use it as a landmark. And then surprisingly for me, more for my supervisor, who got a bit disappointed about the result of nature work. So is that just the animal, when there's such an ambiguity, it will use any available information to know where it is. And also that the activity of these 2 regions is strongly related. So why are these things important? I mean, it's not just for my intellectual curiosity. It turns out that there was a project done by urban designers in London about 5 years ago, and they used some of the data from John O'Keefe to come up with not completely but partially to come up with a system that you can map important locations in London so that you can find your way around very easily. So the way that they came about is there's a scale in the map so that you know how far things are. It always tells you where you are and also where you are in relation to landmarks. And there's another map. I encourage you to see this. It's a work of art. You can also see in 3 dimensions the buildings as you are standing from the map to the external world. So you can update where you are and where you will be heading in a matter of minutes if you can read this map very nicely. Whereas if you use your GPS and it just doesn't update fast enough, you'll get lost or you'll be walking in the wrong direction, which is something that's happened to everyone. But finally, and for me it's more important and more fascinating, is that this bridges together something fundamental in neuroscience, and is how is the sensory information from the external world, auditory, vision, and so on, how is that mapped into the activity of neurons, and how that is generated in internal computations to high sensory processes, memory, perception, in this case, spatial orientation, and so on. So this is my supervisor, Miguel Ángel Valencia. He's helping me with statistics and a lot of discussion. Pierre Vive, who also helped me with co-recording in retrosplenia, which is extremely difficult structure to work with. And Amir, who's been just an amazing postdoc, helping me in all sorts of ways. And thanks for listening.
A little bit into some questions, so we may only have time for.
Okay.
You have one as well.
I can quickly address the 2 questions. But, yeah. All right. So the question about geometry, right?
Yeah, well, I have several, but yeah, you can
All right.
answer.
So the question about geometry turns out that it depends on the degree of disorientation. So if the animal is highly disoriented and it can't use any of the visual information, now it uses the relative geometry of the room to know where it is. But it's a weak effect. That's in the head direction cell system. So basically, if you go from a cylinder to a square, the cell is likely to follow one of the corners if the animal is completely disoriented. It's more interesting what happens with grid cells. And just this year, from the group of John O'Keefe, they published an article in Nature where they show that that if you change the geometry of the environment, you morph the or distort rather the activity of grid cells. So you have shown that grid cells maintain this periodic triangular pattern, right? So when you change the geometry, like if you make a triangle, then the animal, it's very difficult for the animal to compute its position by triangulating points in space relative to corners. So it's easier to compute where you are in an environment that is a lot open. But when you go in a corner, it's more difficult for you to pinpoint your location. So the map becomes distorted.
Okay, can I ask more? So what kind of rat is this? What's the strain?
The strain?
Yeah.
The strain is, I should know this. So these are, Mr. Hooded.
I'm just wondering because if there's a difference between the albino rats and the non-albino rats, because obviously the size, I would suppose it. But you say that if you have no cues, with no cues, with lights on or off, it doesn't matter. They still you still have this firing of the neurons, right?
Yeah. So the activity persists.
Yeah.
And the direction that itself fires begins to drift because there's air that accumulates as you move around in a dark environment.
And I might ask more. So rodents like corneas. So do you habituate, just more like a technical question, do you habituate your animals to the arena and then I suppose you record? Right?
In principle, there's no need to habituate the animals because to get one of these cells, I would have to record more than 300 trials.
Right.
So yeah, so there's no need that.
But at the beginning, because as I said, they like corners, so
yeah.
Probably the first trials are going to spend a lot of time just like around the corners probably not crossing from one point to another.
So
That what happens.
yeah, so this is it gets a bit more technical. So for me, it doesn't really matter because what I care is that the animal faces all the directions, right? So as it moves around, it's facing all the directions just to get good sampling for creating these plots. It's more important for grid cells. So for grid cells, you really want to get good coverage across the environment. So in that case, yes, you need to familiarize the animal and the environment. So Vanessa, last question.
Okay, so yeah, I was wondering, what is the sex of the rats?
We only work with males,
Like, do you think that would
just
be,
like my colleagues who work with males.
yeah, but do you think that might cause an influence, like having female
I don't think it does because this is so. So the way that the brain computes space is fundamental across sexes and across species. Something that I'm saying is that heteroaction cells, some base cells have been found. Well, recently they were found in flies. They've been found in bats. They've been found in squirrels. They've been found in some depends how good the signal is. You found in humans, non-human primates. So this is a fundamental way that the brain computes space. So it's not dependent on the gender. But it's a bit more complicated for other things like with place cells. Place cells are much more variable, and they can be influenced by odor, context, and other things that I didn't address. Right? Thank you.
So, good morning, everyone. Thank you very much for coming here. I'm Andrea Ochoa, and I'm studying a master program at the Division of Surgery and Interventional Science in Nanotechnology and Regenerative Medicine at here UCL. So, to begin with, I would like to ask, do we have doctors or someone who would like to give a shot? So, we have two patients. Patient A has abdominal pain, vomiting, anorexia, weight loss. And patient B has also abdominal pain, but also abdominal distension, jaundice, and dark urine. So,
What is
who would like to
jaundice?
try? Jaundice is yellow pigmentation of the skin and eyes.
Oh.
Yeah. But yeah, so anyone would like to try to see what's wrong with these patients? What's the clinical diagnosis?
They went to my stage last night.
Probably.
Liver failure.
For which one? Patient A or
Patient B.
Yeah. Okay. So, well, both pancreatic cancer. So, as we can see here, these very two different clinical signals is for pancreatic cancer, and pancreatic cancer is a very deadly disease. Only 72% of the people that has it survives 5 years. We can see in this graph that the lines between death and new cases are really close. And even if it's not very common, only 3% of the people that has it is very deadly. So we need to do something with the disease. So why this disease is highly mortal? Because we don't first we can diagnose it on time. There's a poor prognosis of this disease. There are no clear symptoms that we can see. It could be anything. Also, only 95% of the patients die between the 6 months. The current treatments are not good enough. So we need to understand the disease from a different point of view with 3D cultures, ECM. So the current models that we have are mice models, but we need to understand that mice are not human. There are intrinsic differences between the two strains. Also, there's a lack of single genetic pattern, and the complex of the tumor microenvironment makes the even more difficult to treat the tumors. The ECM, that is the extracellular matrix that the cells secrete in order to live, becomes very stiff, very complex. So there's a lack of good models. We have transgenic models, but they're not good enough. They don't have specific cells, the pancreatic stromal cells that we need to work more on it. So for that, we have tissue engineering tools that enable modeling of disease. So we have pluripotent stem cells. So what is a pluripotent stem cell? It's a cell that has the capacity of regenerating all the cells in the organism in the three layers, mesoderm, ectoderm, and endoderm. And on the other hand, we have decellularized scaffolds. So the decellularization process is the removal of cells from the organs in order to keep the extracellular matrix. What is the extracellular matrix? Is the proteins and like the home of the cells that they secrete to live, proliferate, and do all their metabolic activities. So we have an example of a pancreas that is being decellularized until it becomes completely translucent because all the cells have been removed. So my thesis or dissertation project was to develop a suitable pancreatic scaffold for modern disease. So in order to do that, we start with diabetes because diabetes is a widely studied disease. We wanted to create a model that enabled us to differentiate the pluripotent stem cells into pancreatic cells. So more specifically, differentiation in beta cells because we have standardized protocols and is widely studied, but also diabetes, as you have heard before, is a huge problem worldwide. 387 million of people have diabetes in the world, 9 million are in Mexico, and a third of that are here in the UK. And we have two types of diabetes. Diabetes type 1, we don't have, there's a lack of insulin because there's a problem between the immune system and the beta cells. For some reason that we haven't discovered why or how, the T cells from the immune system infiltrate or go into the islets of the pancreas and destroy beta cells. And they infiltrate into the basement membrane, so there's a problem with the ECM. On the other side, we have type 2 diabetes, where due to insulin and high glucose levels in the blood, they make the islets or beta cells to become less functional, less insulin is being produced, and all the amount of glucose in the body is not well processed. So the current treatments for type 1, we have lifelong insulin administration of insulin, that is injections, and islet cadaveric transplantation. The problem with this is that there's not enough donors, and we need immunosuppressive drugs because the immune system is going to respond to other cells. And the type 2, we have drugs to improve the receptors' functions in order to release the insulin or to make it higher in glucose processing. Therefore, we need to understand ECM-related disease such as diabetes, type 1, pancreatic cancer, and many others with better models for the ECM, the extracellular matrix, all the proteins that are around the cells. So we need more accurate treatments. So also the final goal for this is organ regeneration. So first, I would like to explain you a little bit how is the process to generate a beta cell from a pluripotent stem cell. To do this, we identified different cell stages with different protein markers that the cells express in their surface or inside the membrane. And these markers are related to the kind of cells, the kind of differentiation, or the kind of work that they do inside the body. So if we add to a pluripotent stem cell that is over here, activin A, that is just a molecule that we call growth factors, we're going to make these cells to produce SOC17. SOC17 is an endodermal marker from the dermal lineage that gives rise to pancreas, liver, lungs. But at this stage, we still don't have a beta cell. So we need to add more growth factors, or more molecules to modify these molecular pathways. So we have retinoic acid. So in this stage, we're going to have the production of PDS1. PDS1 is a marker for a pancreatic progenitor. So now at least we have pancreas in the name. So we are getting closer. But as you may know, the organs are very complex. So we have different types of cells in the pancreas. We have exocrine and endocrine. The exocrine are acinar cells that produce amylase and other different compounds that are in the blood, and endocrine that are producing hormones. So if we add more growth factors, now we have an endocrine progenitor, and this endocrine progenitor is improved. If you add another component called nicotinamide, we can finally have insulin production and a beta cell. But then you might wonder how the cellular scaffold or this thing that we have after removing all the cells can tell the cell how to behave in order to become a beta cell. So it's having many reports in the literature that the stiffness of the place that are the cells is going to manipulate the cell, because it's not the same to go every night to sleep to a hard surface that in our comfortable beds. It's not the same. It's going to change the cell behavior. Also, different key pancreatic matrix proteins as collagen 1, fibronectin, collagen 4, and laminin are very important. This will prove that in collagen 1, we have a specific peptide called RGD that is going to induce at this stage the cell to become the next cell in the next stage of the differentiation process. Also, we have another proteins that induce cell attachment, and another ones that are important for insulin secretion. So, having that in mind, the hypothesis and objective for my thesis project was that the ECM, the pancreatic extracellular matrix, can have cues or signals that guide pluripotent stem cell differentiation into beta cells. The aim was to develop and characterize such a scaffold, and the objectives were to create it, to characterize it in order to see the ECM preservation, and to finally see the cells and see if we get beta cells. So the objective one was to develop the pancreatic scaffold. I tried 4 different treatments, and with the 4 I got very different results. In here we can see the simple side. We can see this one is almost gone. I mean, it was really fun to work with them because sometimes I lost them because I couldn't see them. And this one is, we still have cells on it. This is under the microscope. We can see that these 2 are more, there's a higher dissociation degree compared with these 2. So for the scaffold characterization was preservation of the proteins. To see the DNA removal means that we don't have cells remaining on them, and the biocompatibility in terms of metabolic activity of the cells to see if they were happy on the scaffold. So here we have a comparison between how a native pancreas will look. We have the collagen 1, 4 from lectin laminin. The blue dots that you see there is called, is a stain is called DAPI, and it stands for the DNA cell nuclei. So we have in treatment 2 that the cells are still there, that are still present. But in treatment 4, we can see that we have still DAPI, and here laminin is gone. It's alpha then we need to introduce the cells into the scaffold. The type of cell or line cell that I use is also a reporter cell line, but it's a red fluorescent reporter cell line under the promoter of SOC17 marker. So that means when SOC17 is being expressed, the cell is going to express as well a red fluorescent protein. So we can track this in vivo because cells shine red. So this is just an example to illustrate how at the microscope we will look at different conditions, the behavior of the cells on the scaffold. So for example, here we have a high expression of SOC17 because we see more red. And in stage 2, we can see that this is increasing because the cell is moving forward the next differentiation stage. Also, we look at the metabolism of cells on the scaffold. We can see that the cells are happier in treatment 2 compared with treatment 4. Also, we did an assessment with H&E staining, that is an histological technique to see tissue. So the H is called hematoxylin and stains for DNA. So we can see here in treatment 2, the cells are repopulating more, the scaffolds are growing more into scaffolds. In treatment 4, they stay just in the edges because the edges were the ones that only cellularized. In the middle was not really good on the other picture. So for the difference in insulin expression, I'm going to process it as well, but at this stage, I'm working on the final results because it takes a long time to process the tissue and the preliminary data. I have to work more on that for the validation. But in conclusion, I can see that the treatment 2 has worked better for creating a pancreatic scaffold with more cell removal and good metabolic activity of cells. So these can be useful tools to work in pluripotent stem cell differentiation in order to model disease such as pancreatic cancer and diabetes, and maybe one day to have a complete organ regeneration that could be more personalized medicine. So thank you for your attention, but my special thanks to my supervisor, Dr. Ashley Boyd, and all the members of the team, the PhDs who are very helpful, my academic tutor, Dr. Gavin Gill, and of course, Conacyt, because without them I wouldn't be here, so thank you very much.
So do you go back to the plot? Why is there so much variance in treatment 2 compared to treatment 1?
Yes, there's a huge variance because you get variance in everything.
But you get a lot when you go back to the
Yeah.
treatment. You get a lot more in
Yeah, there's a high standard
sport
deviation.
too, yeah.
Standard deviation. The first thing, well, among the treatments, even to begin with, there's a lot of difference between the mice age. Supposing that these were same age, I tried to receive the mice in the same age and then to do the same procedure. So there's a difference between the two detergents because to the zero scaffold you can mix between detergents, chemicals, temperature, exposure to detergent, agitation. Also, I'm doing immersion. Some people do perfusion, that is with a complete organ. They just put a system in a reactor and they wash the cells. I'm just doing immersion, so I just put the organ in a flask and agitate. So the effects are because also the detergents have different effects on the proteins. So the first one was more into the surface receptors to detach the cell, and this one just destroys proteins. So maybe this one was very harsh, but yeah, it's still that this has to be standardized, this protocol, so we're working on it.
And about how many data points you have for each treatment?
Yeah, for this was 4, and for although it's a small n sample number because for the RMI, so we can keep hitting the mice. So biological.
Just as a suggestion, I think if you just create a scatter plot instead of the bar graph, then you show the variation, and also you can go back to your experiments and see where each data point lies, and if there were any differences in protocol, I don't know, whatever you did, then you can at least preliminarily, before you carry on doing more experiments, try to identify the source of that variation.
Yeah.
So you're gonna get pancreas empty and then refill that pancreas with your new cells?
I'm doing both things. I get the pancreas, just like here. I get the pancreas. I do the treatment with detergent, agitation, temperature, and then I try to get this, just the ECM, and then I characterize the ECM to see ECM, the protein preservation, and then I put new cells, and in this case was the reservoir protein cells in the cell line, to see, and also I do the differentiation process, adding specific growth factors to see if we get beta cells for insulin producing beta cells.
So to be 100% compatible to the person or the model, it has to be the first cells has to be from the same individual. The pluripotent cell has to be from that.
Yeah, ideally now is the new trend to use stem cells from the same patient and to use the differentiation into beta cells or pancreatic progenitors. So there's no problem with the immune system, and there's no need to use immunosuppressive drugs. So that's the trend. Yeah, that would be really good. But the thing is that forget the pluripotent stem cells, only 2 kinds, embryonic and induced. Embryonic, there's been a lot of ethical concerns because they are embryos, but well, that's a different topic and it's very argued. The IPS is induced pluripotent stem cell. That means that if you can take a cell from your skin, we can put it back into a state of pluripotency, changing 4 genes, and then from there we can go back to differentiation process. But there's also problems with that because with one single cell that remains in pluripotency state can induce teratoma formation, cancer. So there's still a lot of work to do on that to get to that point, but that's the idea, to get all these safe procedures and safe things in order to organ regeneration. But also my personal interest is for disease modeling. pancreatic cancer, diabetes, and
capacity constraints, characteristics.
Yeah, forget that title. That's an awful title. Doesn't say anything. Yeah, so I'm studying cognitive neuroscience, the same faculty as Joey, and I do decision making. And right now, I'm just going to show some of the behavioral data I have from finishing my second year now, the PhD. And I'm going to try to maybe explain what I want to do later on for future studies if we have time. So I want to flirt with some neuroscience techniques called TMS at the later part of the PhD, and hopefully maybe Jave or Lourdes can give me suggestions on what to do next. But basically what I'm going to talk about today is rules of thumb in decision making. So easy strategies that people use to make decisions. And I'm trying to tell you why this is interesting and it's fascinating. So like 30, 40 years ago, David Marr explained that when you're studying cognitive agents and what it's doing in the world, how it's making decisions in the world, you can characterize at least 3 levels of analysis of what it's doing to make a decision. So like on the top level, you can say there's a computational level, which is basically the goal of that agent. What is it trying to do? And what's the best way to do it? And then there's the algorithmic level, which is the level I'm going to talk to you about today, which is what's the program that agent is going to use to achieve that goal in the best possible way? Or how is it going to approximate that goal in the best possible way? And then there's the implementational level, which they've talked about today with my colleagues, which is more the neuroscience level, or the hardware level, like or a computer, for example. What's the actual hardware people are using, or machines are using to carry out a task? And the interesting thing is that all 3 levels constrain the space of decision-making algorithms, right? Because there are infinite decision-making algorithms, there are infinite functions you can use to make a decision, to achieve a goal. So we want to try to reduce that space to see what people are actually doing when they make decisions. So rules of thumbs, another word is heuristic. Why are they important? I'll try to tell you that. And that's more or less what I just said. People, they have a reduced also space of neural hardware they can use and reduced space of cognitive resources. We don't have infinite capacity, not even computers have infinite capacity. So that'll constrain the algorithms we can use to make decisions. And so why not use the optimal strategy? Well, it's because a lot of decision problems are intractable. So for example, chess is a good example, right? You have 10 to the 47 legal positions. Or Go, the game of Go is even more complex. You have 10 to the 171 legal positions. What does that mean? That means that even if the universe was a supercomputer, like if every hydrogen atom was a processor in the world, you would take infinite time, like a really, really long time to compute all possible board game positions. And you have the optimal answer for any position in the board. So this is just a toy example of what I'm talking about. So many problems in the world are intractable. And you need approximations, algorithms that approximate that decision-making process. So even for a machine, if you try to program it to make it go drive to the supermarket, depending on how you characterize that decision-making process, it can be intractable. So you need approximations, you need abstractions to make a problem that seems intractable tractable. So these are examples of what's called empty hard problems in complexity theory. And we're trying to reduce the space of this complexity. And humans do this really well, right? And many cognitive agents do this really well. So there are different approaches to decision making coming from machine learning, artificial intelligence, statistical learning, game theory, expected utility, and normative models of rational choice from axioms of logic and probability, which in cognitive psychology they focus most on. But some of my role models come from all these areas, like John Nash and John Van Neumann, Alan Turing. They've all contributed to this area. So in cognitive psychology and in decision making in general, we make the distinction between normative models and descriptive models of decision. Normative models is what's the optimal thing you can do in a given situation, and what's the best way to do it. So if you were a supercomputer with infinite capacity, how should you solve a problem? Then Herbert Simon, in artificial intelligence, a Nobel laureate in the 70s, talked about bounded rationality and what I was talking about just now. So we don't have infinite capacity. So let's try to describe cognitive agents given their environment and given the capacity constraints they have.
And Kahneman and Tversky, also Nobel laureates, Kahneman, they talked about the heuristic bias programming cognitive psychology, which is basically what I'm talking about today. It's like humans don't have infinite capacity. They have limited resources, cognitive resources. So what are the biases, what are the heuristics we use to come up with decisions? And more recently, Gigerenzer is like the arch rival of Kahneman. He talks about heuristics in a different way. He says, well, heuristics, they're not necessarily rational. So given that we have cognitive constraints, we're ecologically adapted to our environment and to our constraints, to our resources. So he makes the claim that, yeah, heuristics not that people are irrational, because you can see a lot of this in some of the textbooks in psychology, like people are irrational, they're not doing the optimal thing. But not necessarily true, just adapted to our environment, right? And I like this approach. So from the Gigerenzer groups, they came up with a toolbox, general toolbox of heuristics people use. And they fit a lot of data and decision making quite well. Two of the more famous ones are called tallying and take the best. So these are basically approximations to certain decision making problems that we've seen people use quite often. They're normally compared to some kind of optimal benchmark, like some kind of linear regression, some kind of Bayesian linear regression model that would take into account all information in the environment to make the optimal decision. But as we know, people can't normally do this. We don't have infinite capacity to do this in decision-making environments. So to tell you about these heuristics, tally and take the best, I'll just give an example. So suppose you need to choose if you want to eat an apple or not. I don't think anyone wants to eat this apple, right? So just to make the difference between both heuristics, tallying, what it does, it's similar to linear regression. The linear regression has weights for each cube, right? For each variable you put into the regression. Tallying, what it will do, it will just give it a sign. It will just say if it's +1 or -1. And it'll just treat all cues as equally important, and it'll give it a score at the end. So does it have more good cues or more bad cues? So in general, this apple, is it big, is it tasty, is it rotten or not? It'll just give a +1 or -1 to all those cues and add them up. And if it's positive in the end, well, you might choose the apple. Take the best uses much less information. It'll rank all the cues in terms of importance, but it'll just choose the very best cue. So for example, if I'm only interested in the size of the apple, that'll be my best discriminating cue. And if it's positive or negative, that'll totally determine whether I choose the apple or not. In this case, taste, for example, doesn't have either a check mark or a cross, which means that that cue in this given environment is not discriminating anything. It's not giving me any information to try to choose, to try to make my decision. So take the best is a much more frugal heuristic. It uses much less information than tallying. And you would think that a heuristic that uses less information is more irrational, would take less time to compute. That's an assumption many people make. And this part of my PhD is based on trying to distinguish exactly what cognitive resources people are using to implement these algorithms. And yeah, so this is the question: which heuristic should we use, or which one is harder to use? Which one uses more resources? Some of the resources you can think of are the time required to use the algorithm, the information you need, how much accuracy you need in that decision-making environment, how much energy, mental effort, working memory capacity. There are many resources you need to implement an algorithm in a decision-making environment. I'm going to focus more on the time constraints in my paradigm to talk about these algorithms in decision-making. So the hypothesis, even though tallying uses more information than take the best is more resource intensive for humans. Why is this? Is because it's the way the algorithm works, which I'll explain a bit more shortly. And it's because also the way working memory capacity works in humans and how selective attention works, how we attend to different cues in different orders will depend which heuristic is faster and which one is more resource intensive. So the way I set this up is in psychology, we use a lot of cover stories, right? So we just make up a cover story to make up a decision-making environment. In this case, our cover story is, we're going to ask you which country is going to get more money next year, which one is going to have a higher GDP next year. But that's just a cover story, it doesn't matter. We're only interested in how people take information from this environment and how they compute the decision. So in previously other experiments, they just they train people in an environment and they try to deduce what algorithm people are using from the data. You get a binary vector of responses and you try to fit that data with some kind of model, like telling you take the best or linear regression, and you try to see what people are doing. In this case, to take away any of the variability, we're just explicitly telling people, okay, this is the algorithm, want you to use this algorithm for this decision-making problem. So we tell them we train them how to use tallying, or we train them how to use take the best, and then we're just going to compare their performance, basically. So the study design is in 3 phases. That's within subjects, and between subjects is the heuristics I was talking about. One condition is going to have tallying, one condition is going to have take the best. So in the first phase, you practice the rule, so you have perfect performance on the rule. On the second phase, it's a self-paced phase, so we take away some of the information that we gave you to learn the rule, and you have a limited time for each trial to make a decision. And the third phase, this is all counterbalanced and randomized, but the third phase, just to make it easier to explain, has a 2-second deadline for each trial. So you have 2 seconds to make a decision for each trial. You have 72 trials on each phase, and there are different levels of difficulty, depending on the heuristic you're using. So take the best has its own levels of difficulty, tallying has its own levels of difficulty, given the way the algorithms work. So yeah, the first to do the practice phase, this is more or less what they saw. They would see statistics for the countries, and they would have to choose which country A or country B, which one is gonna get more money next year. So if you're doing take the best, you want to look for the best discriminating cue. So here you have the order of how important these cues are. So in this case, this is a hard trial because they have to look all the way to the 3rd cue in importance to make their decision. Why? Because public investment is not discriminating. That's why it has an equal sign. Increased employment is not discriminating. The best discriminating cue here is competitiveness and medium enterprises. So if you're using take the best, you would pick country B. If you're using tallying, you would pick country A because it's 2 greens against 1 red. Tallying just sums it up and it would choose country A. All trials would make this way where they would give differing predictions. Because there are trials where take the best and tallying would make the same prediction. We wanted to fully distinguish them. Yeah?
So who's your population? Because I'm just thinking that.
Sorry, I didn't mention that, yeah.
Maybe some people have no idea what's GDP unless you give them a, whatever, a story. And then some people might not know, or maybe I would think that increased life expectancy is like the biggest indicator, but this is just because I'm ignorant, not because, I'm trying to weigh the.
So first of all, this study was done online with 206 people. So it's a big demographic variability in the United States, given this online platform. Second of all, like you said, we give them a story. We try to explain some of the statistics, but really it doesn't matter what people think about the statistics because we actually give them how important they are. So they're already ranked, and they're explicitly ranked, and all this is randomized. So people will get different rankings for everything. So even if people have come in with a prior belief of which statistic is more important, or they're not really sure what it means, that variability should wash out across all participants because everything's randomized. Yeah, 5 minutes, okay. So they would get feedback to know if they did good on the rule or not. And then once they have near perfect accuracy on the rule, they go to the test phase. Sorry, they don't see the ranking anymore. They just have to remember which cues were more important. For tallying and for take the best, it's more important for take the best. So that's where tallying and take the best differ, right? Because take the best, you actually have to remember the order of the cues. So you need more working memory capacity. You have to have this in your mind to actually make a decision. Tallying, you don't necessarily need to remember that disordering because you don't care. You're just treating every cue as equal importance. So in the second test phase, you have the deadline, and if you took too long, you would say, sorry, you took too long, and that would be a missed trial. So I'll just go through quickly the reaction time differences. What I'm showing here is there's a big difference. Like on average, a trial for take the best will take around 4 seconds in the no time pressure phase. And tallying takes about 1 second. And in the time pressure phase, obviously, tallying would do really well because it's way below the deadline pressure, even though there is a statistical difference between both phases because it's still a time pressure. It's not that effective in terms of reaction time as take the best. I mean, take the best, you can see a massive difference, which is expected, and it's just a manipulation check, right, to see how big a difference we can get. But what's interesting here is I'm plotting the trial difficulty. So the more difficult the trial was, either in tallying or in take the best, you can see we get significant differences. I won't go in too much into the difficulty, but in basic terms, like take the best difficult is like what I said, just how far down the rank you have to search. Tallying, it has to do with how big of a difference you have between greens and reds on a given trial, on good and bad cues. But what's more interesting is the accuracy. So, like I said, even though tallying uses much more information than takes the best, it's a more rational heuristic. It does much well with it. It does super well with this time pressure, and we haven't seen this before in the literature. And take the best, it's just much more frugal. It's massively affected by this time pressure. So you can't really see it here because it's a projector, but basically tallying, you can see that the same distribution for the accuracy. And with take the best, I mean, it goes way down. I mean, it's basically random choosing for the hard trial, the medium hard trial, and the hardest trial. And you have a bit above random choosing, so around almost 60% correct for the easiest trials and take the best, which is basically telling you that under the time pressure, people can only remember the best cue, and that's what they're going to focus on. So take the best is really fragile to time constraints and other type of resource constraints compared to tallying. And it's just a bit counterintuitive, like I said, because tallying uses much more information, but it's doing much better. But we try to give an explanation of how this works later on. So I'm just going to go through this real quickly, but basically you have other models you can test, which is basically using subsets of cues. So if you use all possible subsets as a model of what people are using in the time pressure phase, you can get a bit of extra information. Like I said, people are just using 2 cues, 3 cues, 4 cues, 1 cue. So all possible models comes down to around 13,700 models. But actually, that's double. It's more like 28,000 because you can define cues either in terms of their names or in terms of the position. So I can use the 2 best cues or I can use the 2 first cues. So in the end, what we did is we compared these 28,000 models for every subject. The problem when you do so many model comparisons is you can overfit the data easily. So we had to cross-validate. We used 5-fold cross-validation for all these models. And in the end, we just divided them into classes. So the classes of models, we either term featural or positional, depending on what I just said. Are you using the first 2 cues on every trial, or are you using the 2 best cues on every trial, or any arbitrary number of cues? There were only 7 cues in total, so that's finite, right? So for tallying, we got what we expected, right? I mean, it's either something called a positional tallying model or a featural tallying model, which we can't really differentiate between the two. But basically what it's telling you is people are using practically all the cues in both phases. And it's really irrelevant if you're tracking the names or if you're tracking the positions of the cues. And it's also really hard to disentangle this, especially when you're using all cues. There's really no way to disentangle if it's based on the cue names or on the cue positions. You would need a new design to try to disentangle these classes of models. Take the best, we also got what we expected. Which is basically, take the best, you have to track the names of the cues, because otherwise they won't work. And I'll show you a plot of what cues are actually using for take the best. It's hard to disentangle this with what's something called featural tallying model, because it's just the way we designed the environment so that every trial would either be take the best or tallying, which makes it that you can do a complementary strategy that would give you a very good fit to what's called featural tallying. But this is just an artifact of the environment, but it causes a problem for the analysis. And this is just for the winning model and take the best. It's just showing you that, people are actually using the best cue more often, the second best cue more often, third best cue more often. And these are practically the same because they don't matter that much. I mean, we didn't have any trials where you had to look for the 4th, 5th, 6th, or 7th. So just summary of results, take the best, even though it's a more frugal heuristic on tallying, it requires more computing time because of its search strategy. Because it's a serial search strategy and you have to go 1 by 1 cue to see which one is the best and which one has information. This is partly supported by the subset modeling I just showed you. I didn't talk about the effect of non-discriminating cues, but that has an effect on reaction times. Cues that don't have any information for a given trial, they'll affect your reaction time and your accuracy for take the best, but tallying is not affected by these type of cues. And for future studies, like I said, we can use information from neuroscience techniques to try to constrain our algorithms further. So I'm not too sure on how this design is going to work, but Anyways, our hypothesis is that there's an attentional mechanism that's necessary for take the best, that's not necessary for talent. And then we want to test this is to knock out an area, which is called left dorsolateral prefrontal cortex, that has been highly associated with processes of cognitive control. But specifically, we want to make the claim that cognitive control is necessary for a selective attention mechanism that you need in order to use take the best. Whereas tallying, you don't need this selective attention mechanism given our design. And yeah, so just some take-home questions. How do different heuristics perform under different resource constraints? I'm interested in what are the psychological mechanisms that account for these differences, like selective attention and cognitive control. And specifically, we want to see which brain areas are necessary to carry these psychological mechanisms. And I'll just give thanks to my lab, my supervisor, and classmates. Thank you. Yeah.
Okay, so sorry, I'm not taking notes and I forget the question. So you have these two strategies, and when there's no time constraint, both strategies are equally effective.
Yep.
But in a situation where there's shortage of time, tallying is the best strategy.
Yes.
So how does these two strategies relate to what Kahneman and Tversky investigated for many years with this slow and fast system?
The slow and fast system.
Yes.
Oh, interesting. So I think what they were talking about there, for also people who don't know about what they were talking about, they were talking about an explicit reasoning system, which is what people think of as maybe conscious decision making. So when you consciously manipulate information to come to a decision, like solving a mathematical equation, solving any test at school. So that would normally take more time and is slower because you're manipulating symbolic information, as opposed to intuitive decision making, which you can also use to maybe solve a math question where you don't explicitly reason through a decision, but you just intuitively make a decision and you intuitively come up with an answer, which is much quicker. So when Lourdes is asking, how does this relate to this? Very interestingly, yeah, I think it can relate a lot. So the nature of the task is very visual. So you can see for tallying, you can just do some kind of visual integration to come up with an answer. And you can say that's an intuitive strategy. You're not explicitly manipulating some type of symbolic information. So you can just use the visual information environment to come up with a really quick decision. And obviously visual information is processed much more quickly than any other type of information, like actually trying to process the names of the cues and going one by one and getting the information one by one, which is what Take the Best is doing. So that's also what we're talking about, the psychological mechanisms here. We don't want to just say, oh, there's a fast system and a slow system. No, okay. What actual psychological mechanisms do you need for each strategy? And why is that giving you a difference in reaction time? So I think that sort of answers your explanation. I wouldn't want to say there's a slow system and a fast system. There's processes that are faster in the brain than others.
But I would assume that for both strategies, you must need the cortex to, if you're talking about which structures in the brain are related to this, I would think that for both, you would need the cortex. So,
yeah, I mean, definitely, because if not, you couldn't even understand what the task is about, right? I mean, but what we want to make the claim is that you can selectively knock out an area of the brain, for example, like left field PFC, just that area, to knock out just cognitive control for selective attention. And that's a very specific process that we're speculating that's important for Take the Best.
So you would also promote impulsive choice.
Right, right. Yeah, so, but that would reflect itself also in error rates, wouldn't it? If it's just impulsive choice, wouldn't it
Well,
just...
not necessarily.
Uh-huh.
Not necessarily.
Yeah,
I mean, because there are similar. Obviously, I work with rodents in similar tasks, and you won't necessarily see more errors or omissions. They would just, choose the non-optimal option, which is not necessarily an error. It's just, a bias or a change of preference.
yeah, with changes when it's subjective value based decision making, it's a bit more difficult. it's hard to say what an optimal decision
Yeah, that's
is. And here it's really clear cut what an optimal decision is. I'm not sure how it relates to your type of paradigm. But yeah, I agree. I mean, if you knock out an area that's related to cognitive control, it's generating more impulsive behavior. It's generating also better moods. I mean, a lot of the treatments they do with this kind of technique, which is called transcranial magnetic stimulation, they're using it to treat depression now in exactly the same areas I'm trying to target. So I don't know, maybe you can just do better because you're feeling better. But I think that would be a crazy explanation. But anyways, yeah, it's a confound that I would have to deal with for the future stuff.
Just a quick question. So can you speak more about the ecological validity of using heuristics when there's
So like what Giger is talking
yeah,
about?
so when there's a short and long term long time constraints for your decision, and I'm thinking more of so it's like fundamental biology. If you think of natural selection and especially sexual selection, the decision either for identifying a potential mate or, in contrast, to identify a potential predator has to be done within seconds.
Within seconds to
Well,
choose a mate.
Well, it depends how. Well,
You're fast.
yeah, I was thinking more about how the brain would actually make the decision, but the decision itself within minutes. Whereas with humans, that might be take much longer. So the idea here is so the idea is like if you translate that to the idea that we are somehow irrational when we make these decisions that might seem suboptimal. Thinking when you have all the evidence and it's being weighted across the long term, how does that actually compare between exactly those two models that you were saying from Gier and Kahneman? And in relation, especially to your data, where you show that if you limit the time constraints, then you basically don't see that change in the strategy.
So I'm not sure I understand your question. So why is it ecologically rational? Is that what you're asking?
Yeah.
I think
Or irrational, I mean, depending on which of the
I really hate the term. I hate the term. I mean, I have to describe it because that's how the field grew up. But it's a straw man. It's something people use as a benchmark because it's the optimal behavior given some function. But the function you're optimizing can be arbitrary as well. So I really just think it's a straw man for these people to debate about. But it doesn't really matter if you call it rational or not. I mean, I think if you put more resource constraints into your model, you're making the model much more realistic, which is what we're trying to do in this type of paradigms. And it's much more descriptive of what people are doing or can actually do. It just depends how detailed you want your model to be, your model of decision making to be. And obviously, if you put time constraints in there, or if you put memory capacity in there, you can do the same thing for a computer. Like people in computer science, they do the same thing. Okay, which algorithm is going to give me the best accuracy I'm willing to accept, or my company is willing to accept, in the shortest amount of time? So that's a different objective function you're using right there, because you don't have infinite time, so you have to optimize this objective function.
So in principle, you can run your experiment as like long-term design, and then get some kind of function where you can actually see how the decision is weighted from limited information to more information, and then
Oh yeah, definitely. That would be crazy interesting.
Yeah, so kind of like psychophysical curve, and then just characterize. Okay, so what is how's the function behaving?
Definitely, in principle, you can do that.
Yeah.
Correlate amount of information.
So that would be like very powerful.
Yeah. No, so it's a really cool idea. I just have to think about
which is done a lot in psychophysics for discrimination testing, like when you do the electrophysiology.
Yeah, to get a psychometric curve.
Yeah.
Oh, that would be really cool. That's fine.